Next is chromosomal segment substitution lines or CSSLS. Chromosome segment substitution lines represent a powerful method for precise quantitative trait loci, detection of complex agronomical traits in plants. CSSL are a series of inbred lines produced by crossing and backcrossing the donor and recipient parents by using marker assisted selection or MAS that finally contain the entire genome information of the donor parent. Each CSSL carries one or more donor chromosome segments in the genetic background of the recurrent parent, thereby eliminating genetic background noise and allowing the detection of QTL with additive minor effects that are always masked in F2 or RIL primary populations. In addition, development of CSSL by using MAS has provided a new breeding strategy for the improvement of cultivated species and allowing the introgression of novel genes of alleles from wild relatives. This strategy has been successfully applied in various crops. Furthermore, the use of these CSSLs has enabled the accurate identification of QTL that govern complex agronomic as well as allowed the detection of the interaction between QTL and multi-environments. The next is Immortalized F2. The F2 generation of a cross between two either inbred or outbred lines is suitable for analyzing various QTL effects including imprinting effects because it contains all possible genotypes at a locus with two different alleles, including two different homozygotes and two reciprocal heterozygotes. The outbred F2 design is most convenient for outbred species. The inbred F2 design is convenient for inbred species and also applicable to outbred species. In this design, the origins of alleles at informative marker loci possessing more than two alleles in the F2 generation can be traced back to the F1 parents and the founder grandparents. Therefore, it is suitable for genome-wide mapping of IQTLs. For complex traits, some quantitative trait loci or QTLs may also exhibit imprinting effects, that means show different genotypic values between reciprocal heterozygotes and hence are termed as imprinted QTLs or IQTLs. The parental origins of marker allele in the inbred F2 generation cannot be directly determined because the F1 parents are identical genetically. Nevertheless, based on the variation of recombination rate between different sexes, the parental origins of haplotypes can be distinguished and therefore IQTL mapping can be still performed. Next is the advanced intercross lines or AIL. An advanced intercross line can provide more accurate estimates of quantitative trait loci map location than conventional mapping populations. And AIL is produced by randomly and sequentially intercrossing a population that initially originated from a cross between two inbred lines or some variant thereof. This provides increasing probability of recombination between any two loci. Consequently, the genetic length of the entire genome is stretched, providing increased mapping resolution. Next is the recurrent selection backcross populations or RSB populations. Backcrossing is a well-known and long-established breeding scheme where a characteristic is introgressed from a donor parent into the genomic background of a recurrent parent. The characteristic could be a trait, a gene, or even an anonymous locus or chromosome segment. Backcrossing is also useful to dissect the genetic architecture of quantitative traits because it isolates a gene or chromosomal region in a different genetic background, that means the genetic background of the recurrent parent. In fact, it is one of the few reliable methods to validate the additive effect of quantitative trait locus or a candidate gene. In addition, backcrossing could be used prior to or in conjunction with QTL detection to increase the precision of QTL mapping. Selection in backcross programs is used to either improve the genetic value of plant and animal populations or fine map quantitative trait loci. Both cases are helpful in our understanding of the genetic basis of quantitative traits variation. In successive generations, progeny are selected for the characteristic of interest and then backcross to the recurrent parent. Backcross and introgression are useful for genetic improvement in breeding programs. The next is types of recurrent selection. There are 4 types of recurrent selection. The first one is symbol recurrent selection or SRS. A type of recurrent selection that does not include tester is referred as symbol recurrent selection or phenotypic recurrent selection. The second one is recurrent selection for general combining ability or RSGCA. A form of recurrent selection used to important the general combining ability of a population for a character, and the heterozygous tester is referred as RSGCA. It is also known as half sub-recur selection. The third one is recurrent selection for specific combining ability or RSSCA. It was originally proposed by Hull in 1945, a form of recurrent selection that is used to improve the SCA of a population for a character by using homozygous tester is referred as RSSCA or recurrent selection for specific combining ability. It is also known as half side recurrent selection with homozygous tester. The fourth one is reciprocal recurrent selection or RRS. A form of recurrent selection used to improve both GCA and SCA of a population for a character using 2 heterozygous testers is known as RRS. It is also known as recurrent reciprocal half SIB selection. Interconnected populations. Interconnected populations can lead to an increase in heterozygosity, which is beneficial if recessive deleterious alleles are masked or if heterozygosity is of a general fitness advantage. Theory predicts heterosis in crosses between individuals from populations that are connected by some level of migration.